The overall goal of this experiment is to label mitochondrial DNA replication in Drosophila adult ovaries by EdU staining.
This method can help answer key questions in the mitochondrial field, such as the process of mitochondrial biogenesis and mitochondrial DNA inheritance during development. The main advantage of this technique is that it allows for good structural preservation and efficient fluorescent dye penetration of whole mount tissues.
To begin the experiment, take a vial with Drosophila medium containing dry yeast and culture 10 adult female flies with 10 adult males at 25 degrees Celsius for 2 to 3 days. Ensure flies remain well fed throughout incubation. Anesthetize the flies on a carbon dioxide fly pad and pick female flies with a paintbrush. Add several drops of room temperature Drosophila medium supplemented with 10% FBS to a dissecting pad and place it under a stereo microscope. Using sharp, fine-nosed forceps, grab a fattened female fly at the lower thorax. Keeping all tissue immersed in medium, with a second pair of forceps, tug gently at the extreme posterior of the fly until the tissues in the abdomen are exposed. Next, detach the 2 ovaries from the surrounding gut and extraneous tissue. Open the ovaries by pulling gently and expose the ovarioles. To aid penetration of the reagents, pass the tips of the forceps between each ovariole a couple of times. Finally,